one of the best PhDs in India. And after getting his PhD, he joined Syngenta Seeds, and he worked there for 2 years. And then in 2006, he joined ICRISAT. He's with ICRISAT for the past 15 years, and he's currently working as principal scientist, genomics.
Sandeep, please mute yourself, Sandeep.
Sandeep Sable, could you please mute?
Please mute yourself.
Thank
Yeah,
you.
thank you. Yeah. Go ahead.
He is currently working as principal scientist at ICRISAT, handling genomics, pre-breeding, and bioinformatics. He leads the Pearl Millet Genomics and Molecular Breeding team at ICRISAT. He is extensively involved in the generation of cutting-edge genetic and genomic resources, mining, mapping, and deployment of QTLs and other genes for various biotic and abiotic stresses, grain and fodder quality related traits in pearl millet, which is also ICRISAT's global mandate. He led a team comprising of national and international partners to define heterotic gene pools in pearl millet for the first time. You know, those of us who are involved in seed industry, we all work with hybrids all the time, and you know how important heterotic gene pools are for us. And this concept, which came up first in maize, is now getting extended to other crops. And very happy to see that Rakesh has done work on heterotic gene pools in pearl millet for the first time. And if they can get the molecular basis of heterotic gene pools, that will be something which could be transferable across crops, I suspect. And it's a very useful work, especially for seed industry. He also led the pearl millet downy mildew genome sequence. And we know how important downy mildew is. It is the, you may, as a breeder, one may produce the best of the pearl millet hybrid, but downy mildew is the one which takes it down over a period of time. So adding stability to yield is contributed by downy mildew resistance, and his work in this area is extremely important. He has contributed significantly to pearl millet genome sequence work, including the development of resources such as World Association Mapping Panel, representing the global pearl millet genetic diversity and mapping populations. He is currently working on the development of new genetic stocks, which is part of the pre-breeding work, functional genomic resources such as tilling, trait mapping, especially for traits like iron and zinc levels, low glycemic index levels, and nitrogen use efficiency, fertility restoration, striga tolerance. I think that's a very important work that you are doing, because striga is a problem which is not recognized well. I have seen in pearl millet also it's a problem in many countries, and on low phosphorus, response to low phosphorus content, and deployment of these traits in elite genetic backgrounds. So he's a breeder who is into molecular biology and focusing on traits which are of extreme importance to the plant breeders and also the seed industry. Some of the key translational genomic outputs from Rakesh's team include the mega pearl millet cultivar HHB67 improved 2, which is now grown on more than 850,000, close to 1 million hectares every year. If you look at this, close to 1 million hectare out of 6.6 million hectares of pearl millet in India, that is a very significant contribution by a plant breeder who is specialized in molecular biology. And also Maru Sona, the first molecular breeding product to be released by state of Gujarat. And he has over 100 publications, including journals like Nature Biotechnology, which is a leading biotechnology journal. And we from ATPBR, we are very grateful to you, Rakesh, for sparing your valuable time. And like Aparna said, we try to connect the academia and then especially students and practicing plant breeders who are young and energetic and bright, and they're looking forward for a good career in plant breeding, and addressed by you I'm sure will benefit them a lot. So I now hand you over the platform, and I request you to deliver your talk on translational genomics in Pearl Millet Advances and Prospects. Over to you, Rakesh.
Thank you so much, Dr. Rao. I'm so overwhelmed for such nice introduction. That too coming from my senior ICRISAT colleague. You've been kind of lighthouse for us. We have read your papers, we have seen your work, and we take a lot of things out from the good work you have done at ICRISAT, including wonderful things you have done on Striga in sorghum. So, sir, this is really a great privilege for me. I'm really humbled to be introduced by you, a scientist of your stature, and also my thanks are due to ATPBR for providing me this platform and this opportunity to connect to so many diverse audience, especially students here. And my special thanks to Dr. Tiku, who I'm not sure many of you would not know about this. Dr. Tiku was my first employer. In fact, he interviewed me for my first job, and I still remember those questions and answers and the discussion which actually happened in that interview room way back almost 18 years. It is fresh as pristine breeze in my mind. So Tiku sir, thanks very much for all the help and support, because that was a primer. I was fresh from IRI, and you actually gave that solid foundation to connect this genomics with downstream molecular breeding technologies. So thanks very much to you, and also thanks very much to Aparna. Aparna also had a brief stint at ICRISAT, so we all have some commonalities. So thank you very much. And now allow me to proceed with my presentation. Just give me a minute. I'll just start sharing my presentation. So please confirm if you can see my slide.
Yeah, we can see.
Hear me loud and clear?
Yes, we can. Yes, we can.
Thank you very much. So in the next 45-50 minutes, I will take you through what it means in Pearl Millet Translational Genomics. It's a wonderful crop. So I would just give a kind of a brief overview of the crop and then take you to the genomic technologies and various advances we have made in this particular crop. So just to set the platform at the right stage. So we all would like to know why genomic technologies. I mean, plant breeding has been doing fantastic in various crops in the last 5 or 6 decades. But then why do you need genomic technologies? So it is for the simple reason that, unless and until you bring in innovation on the table, you cannot actually take a jump when it comes to yield and other things. We have seen yield plateauing in many of the crops. So unless and until we bring in cutting in technologies, there's no way we can actually take the yield to next levels. So of course there are many technologies which have potential to take the yield to the next level. But today in our talk, I'm going to talk specifically about genomic technologies. And this technology is especially important in current scenario because we all have seen how climate change is wrecking the entire globe. We are seeing desertation and marginalization of the arable land, and on other side we have this burgeoning human population, which is like human atom bomb, which can explode. It is like the Malthusian catastrophe, which is actually waiting to explode. And by the current, so as of now, I just downloaded the current population of the world. So we are at 7.9 billion. So this is a staggering figure, and we will be more than 8 billion by 2025 and more than 9 billion by 2050. So to support this huge population, we need to produce more, and we need to produce more in a sustainable manner. And genomic technologies can definitely help. Of course, there will be other technologies which also would be needed to make it sustainable. And if you see the other side, I mean, these many people sitting here on this planet, we need a lot of resources to support ourselves. You see the demand for energy, the demand for power, the demand for all inputs, it's increasing, and this is actually not pretty sustainable. And even at current world population of 7.9 billion, we need about 1.7 planet Earths actually. So this is through some simulation study to take care of the waste we are actually generating, especially plastic waste and toxins we are generating through our various activities. And so we look on to these next generation technologies to take us out of this difficult scenario. So before I get into Pearl Millet, let me also, for some of you who do not know about ICRISAT, let me tell you briefly about what ICRISAT is. So ICRISAT stands for International Crop Research Institute for the Semi-Arid Tropics, and we are one of the CG centers. Of course, one CG stuff is happening, so I don't want to get into details of that. So though ICRISAT has not joined one CG, but it is still part of CG consortium as for now. So ICRISAT actually works on 6 very important crops, which are specific to specific environments where many private companies especially have not ventured in. So these 3 crops include 3 cereals. Pearl millet, the crop I'm going to talk about, sorghum. Dr. Rao worked on this wonderful crop, and finger millet was the latest addition we had. Apart from this, we also work on 3 legumes, chickpea, pigeon pea, and groundnut. So these are the 6 crops ICRISAT actually works on, and it also maintains germplasm of all these lines, including all minor millets. And we actually have our work which is spread in about 55 countries and covers about 6.5 million square kilometer area. And it touches life of this slide is told more than 3 billion people, globally. And you are actually most welcome to visit this beautiful campus. This is a bird's eye view of ICRISAT campus. So this campus has got precision soils. So on one side of the road you will see black soil, and other side you will see red soil. So in the same campus you can actually carry out a lot of soil-based experiments. So this is a wonderful place, not just from the point of view of scientists, but also from recreational point of view. So I welcome all of you to come down with your families. ICRISAT has many lakes, and some of the lakes would house these migratory birds coming from different parts of the world for nesting. So it's a beautiful place in terms of science and in terms of recreation. So all of you are most welcome. Now, coming to the crop in spotlight today, pearl millet. So you might all know pearl millet. It's called bajra in Hindi, and it actually can grow as tall as me. So I'm just sitting. When I stand, I'd be more than 6 feet. But this crop can grow, more than 10 feet tall. Some of the especially forage type, plants. And this crop is now projected as crop for the future. We talk of climate change, we talk of deep space exploration, wherein the crew has to survive on nutritious food for a long period of time, which actually can be generated using minuscule resources. So pearl millet is considered as one of the crops for the reasons listed here in this particular slide. So amongst cereals, this is the crop which actually touches the boundaries of typical cultivation practices. So they say if you cannot grow rice, you grow wheat. If you cannot grow wheat, you grow sorghum. And they say if you cannot grow sorghum, you grow pearl millet. And then they say if you cannot grow pearl millet, maybe you'll not be able to grow anything else. Because this is what pearl millet is in terms of its plasticity to various abiotic and abiotic stresses, especially in terms of abiotic stresses to heat and drought. So these are 2 key traits on which climate change would be hitting us, and this crop really is an astonishing crop when it comes to these particular traits. So in terms of heat, this crop can germinate at soil temperatures as high as 64 degrees Celsius. So other cereals and other species, the seed will still simply get destroyed beyond 50-52 degrees Celsius. But here, this particular crop can germinate at soil temperatures more than 64 degrees Celsius. You can push it even to 68 degrees Celsius, 4 to 5 hours of this treatment, and it can still germinate. So this is one winning trait which can really take this crop as the choice of crop for climate change regimes. So this is about seedling stage heat tolerance. In terms of flowering stage heat tolerance, this crop can set seed in air temperatures more than 42 degrees Celsius. So many of the cereals like rice and wheat, they will fail to set seed when air temperature rises. We know what happens to wheat when there is a 1 degree rise in temperature. The grain yield could go down by even 10%. Same thing in rice. If air temperature rises 38 degrees Celsius, then there is sterility and there is no seed set at all. And also in terms of drought, by its per se mechanism, it's a drought tolerant crop per se, but also because of its short duration. So this crop actually can complete its duration within 3 months. Most of these single cross hybrids and the early cultivars, they mature well within 3 months. So in fact, 3 months is considered as late segment. In pearl millet, the early segment is around 65 to 70 days. So this crop actually completes its life cycle pretty fast, has got ability to survive in low fertility conditions. So if you see the soil of areas like West Africa and even in India, in Rajasthan, so the aridity soil, so the desert soil would be extremely low on nitrogen and phosphorus. And if you grow anything else, it will simply not survive. It will not give you economic yield. Whereas pearl millet is one which actually can support not just human beings but also livestock. So this has got that amazing property. The other most amazing thing about this crop is it has got extremely high day productivity in terms of its biomass. So this crop, as I mentioned, just completes its life cycle within 85 to 90 days. And within this period, it can produce biomass on the other side of 55 tons. And grain yield on other side of 4.5 to 5 tons. So this actually is more than about 90 kgs of dry matter. If you dry it to 10%, 90 kgs of dry matter per hectare per day. So this is something which actually is not seen in many other crop, especially in cereals. And in terms of its adaptability, this particular crop is amazing in terms of its plasticity. So it can grow in both acidic as well as alkaline soil. So this crop can match finger millet in terms of its adaptability to acid soils, and it can match barley in terms of its adaptability to alkaline and sodic soils. And then coming to its traits related to humans, it is nutritionally dense. So you might know Government of India actually designated all these millets as nutricereals. So this has got amazingly high degree of micronutrients, especially iron and zinc. If you see the background iron and zinc levels in rice and wheat, it's just about 15 to 20 ppm. Whereas in pearl millet, the background variability it starts from 35 to 40 ppm. And there are lines in our breeding team, for example, Dhan Shakti, which have got 85 ppm iron. So these are all iron and zinc rich cultivars. And Dhan Shakti is one example, but there are other lines where iron and zinc actually clocks on the other side of 110 ppm. Similarly, apart from iron and zinc, it has got other important micronutrients, especially chromium and manganese. So these two are extremely important for carbohydrate and fat metabolism. If you see nowadays, the incidence of diabetes is rising so much, so that India has become almost like a diabetes and hypertension capital of the world. So that way, this particular crop can really help. It is also anti-hypertensive because of its high magnesium content, and it is anti-mutagenic. So nowadays you might know the cancer earlier was thought to be a disease of the West. Indians were relatively resistant or immune to it, but nowadays cancer is catching up even in India. And I'm not sure if you got a chance to have a look at one newspaper article which appeared about 8 months ago in Times of India, which projected 5,500 people terminally ill, stage 4, stage 5 cancer patients in Hyderabad alone. So this crop can also help in protection against cancer, because it has got a lot of free radical quenchers thanks to its high polyphenol content, which is present in the grain. And apart from this, it has also got high folic acid, folate. So folic acid is one principle which is actually lacking in most of the cereals. It is present in very low concentrations, whereas in pearl millet it is almost 10 times more than what you get to see in rice and wheat. And this becomes especially more important in West Africa, where food basket is narrow, and more especially for the pregnant mothers. You may know, during first 3 trimesters and last 3 trimesters of pregnancy, doctors give folic acid supplement. So that's possible in India, but in Africa, this crop can really, plug in that gap because of its high folic acid content. And other interesting thing about this is, pearl millet in US is used in producing something called as designer eggs. So the birds which are fed on, pearl millet feed produce a higher proportion of, good fat, which is HDL compared to LDL. So the HDL-LDL ratio gets skewed in positive direction. And these are called as designer eggs, and they are, being sold in the market at premium price. And apart from this, it is a very good quality fodder crop in northwest India. In more than grain, many times it is grown as a fodder crop because of its ability to regenerate and its ability to put up high biomass in a very strong period of time. Now coming to global pearl millet distribution. So this crop is basically distributed mostly in West Africa, which is the center of origin of this crop, and also East and Southern Africa, I'm sorry, and in India. And it is also present in small acreages in other important continents like North America and South America and Australia and Middle East. Now coming to the more specifics of this particular presentation. So, genomic technologies we've been talking about. So we need genomic technologies because the face of plant breeding is changing. This can very well be depicted by the iceberg which we see here. Can you confirm if you can see my mouse? Can you see my mouse? Can somebody confirm?
Yes,
Yes, we can see.
Okay.
Rakesh, there's a request.
Yeah.
There's a request from Dr. Karakwal.
Mm-hmm.
That your audio is not very clear, and if you can go a little slow, then it may become clearer.
Okay, because I'm on a high-speed internet connection. I plug in LAN because I thought maybe Wi-Fi connection would not be stable. So I'm not sure, but others are able to hear me properly?
Yeah.
Yes, sir.
Yeah, it's very
Yeah. Maybe there is some issue at Dr. Karakwa's end.
Rajiv says audio is clear.
Yes, I can hear you loud and clear.
Yeah, thank you so much. And Dr. Karakwal, namaste and good morning. Thank you very much for joining in. You're my teacher. Very nice to have you here, sir. So I'll try to go a bit slow. I was talking about this changing phase of plant breeding. So this can very well be depicted by this iceberg. If you see iceberg, only 10% of the iceberg is visible, and 90% actually is floating down under the sea level. And in conventional plant breeding, we were just targeting the things which were floating on the top side of the water level. Which was visible to eye, which was called as plant breeders' eye. Plant breeders in the past have done a wonderful job, and you all great plant breeders are here, including Dr. Tikku, Dr. Rao, and Dr. Karakwal, and many of you. So that is something which definitely is something which is individual specific. But nowadays, what is happening is all this is transferred to machine. So that means, by using data intensive, stuff. Anyone can do it. It's like, when a plant breeder leaves, that plant breeding program dies. Now here, in today's technologies, it is so much data driven. It is so much on the basis of different quantitative parameters that even when a particular person leaves, of course, human beings can never be replaced. The program does not suffer much because much of the things are being taken care by the huge data, big data, which is being generated, which feeds into this molecular building platform. And this actually helps us to take a deep dive and dig out those small pan-genome additive effect loci, which are spread all across the genome, which are of small effect, but if you actually put all of them together, they actually make close to 90% of the phenotypic variance you see for most of the traits. And hence, you can actually make faster genetic gains. So with help of these, artificial intelligence and machine learning, you can actually go on building your algorithms, go on building your pipeline, and you can make your process more efficient with every passing iteration. Now coming to this crop improvement practice, I just thought maybe I should take this wonderful piece of figure from Vasudeva et al. 2020, which appeared in Current Opinion in Plant Biology. So to do this, you need to understand actually your germplasm. It all starts from germplasm. If germplasm is well characterized, then you go to the genome level, and from genome level you go to the genes, and from genes you go to gene editing and genomic breeding. So these are the 5 Gs for crop improvement. And of course, in today's presentation, I'm going to talk about the 4th G, which is genomic breeding. Now coming to pearl millet. So pearl millet was considered as an orphan crop in the past because, there were a handful of labs across the globe working on this particular crop. And the amount of funding which actually went into this crop was extremely low. In fact, I have seen myself, and with due respect to the people out there on the funding side, there is something called as big crop syndrome. So whenever proposals are submitted, so obviously proposals coming from rice, wheat, and corn, they attract attention of these donors because these are mega crops. But when it comes to these so-called minor millets, they take a back seat. But now things are changing. 2023 is going to be International Year of Millets, and also Government of India has tagged millet as a nutri-cereal, and there is a lot of actually interest coming up on the side of both consumer as well as the researcher side. So this crop is picking up, but then in the past, this crop had very minuscule genomic resources. Until it was year 2017 when we did this 1,000 genome sequencing work. So this was a global activity led by Dr. Rajiv Vasudeva at ICRISAT, who now has moved to Murdoch University, Australia. So we had all the major players in this particular field from across the globe, and we did not just one genome but 1,000 genome resequencing work. And we used one line which is called as TIF23 due to B1 as the world reference germplasm, which was sequenced at 300x depth to serve as world reference genome. And along with that, we resequenced a lot of lines. So these lines, the first line is the world reference line. It's called as TIFT. It comes from Tifton, Georgia. And this line is the mother line, which actually led to development of a lot of seed parents around the globe, including all those we get to see here in India. So apart from that, we resequenced PMI gap. It's called as Pearl Millet Inbred Germplasm Association Panel. It is World Diversity Panel. I will talk more about this. So 345 lines were resequenced, whole genome resequenced at about 2x coverage. So you might wonder why coverage is not very high. It's for the simple reason that the genome size of Pearl millet is huge. It is about 1.8 GB compared to just about 350 for rice. So the amount of resources which it takes to generate an equivalent X coverage in rice would be many times higher when it actually comes to Pearl Millet because of its higher genome size. So apart from this world diversity panel, we also resequenced wild accessions. We resequenced parental lines of the mapping population, and we also resequenced these BNR lines coming from the breeding program led by Dr. S.K. Gupta at ICRISAT, and also breeding programs representing national partners and some private companies, including Corteva. It was pioneer then. Now, coming to the distribution of SNPs and indels. So when we did this resequencing work on all these lines and also attempted high depth sequencing work on the reference germplasm, so we actually ended up in about 8 pseudomolecules representing 7 chromosomes of Pearl Millet. So this PG1 was something which was not grouped, just like we get to see in genetic linkage mapping. But otherwise, all the 7 pseudomolecules were anchored nicely, and you can see the number of SNPs. I don't want to get into the details of all that. So the total number of SNP was about 29.5 million, and number of indels was about 3.8 million. So this is huge, compared to what we had here, where we can count the number of markers actually on our fingers. And then we had these SNPs coming on this PMI gap world diversity panel lines, which was on the other side of 29 million, the indels on the other side of 3.8 million. And on the parental lines, the mapping population parents, it was on the other side of 12 millions, and when it comes to SNPs, and about 1.1 million when it comes to indels. Now, so this was in 2017, but now what we are doing is we are trying to develop not just gold, but a platinum standard genome assembly. So we have joined hands with Corteva Agri-Sciences, and we are trying to improve the assembly of TIP23-B1, the line which was actually resequenced here, which we did here in this particular exercise. So we want to improve its quality, and now we have chromosome level assemblies. So we have done some high C, and we have got hold of chromosome level scaffolds. So now the assembly has been improved to platinum standard. In addition to this, we have also now introduced 2 more lines, because if you just have one reference line, you run into something called as ascertainment bias. So to get over this ascertainment bias, we have gotten 2 more lines. So these lines represent 2 heterotic pools. The R line pool, so ICMR represents ICRISAT R line. This is 06777, and the other one is again ICRISAT's B line, which is called as 843B line. So these lines are all again founder lines for the modern breeding program. I mean, if you draw a graph and see how these lines are actually pitched on time scale. So TIP23-B1 actually falls behind, whereas 06777 and 843B, they fall very much into the current breeding program. So if we have a high quality genome assembly, it will definitely help in genomic selection and other important works related to breeding. So this work is almost completed, and we would soon be putting a paper out. So work is going on in the background. Now coming to something which would be useful to the Pearl Millet breeders and molecular breeders, globally. So we developed something called as a virtual high density SNP array. So we started with about 32 million SNPs, and then we filtered it down using various filtration criteria. And we came down to about 68,000 good quality SNPs, which form as a very robust hap map for doing GWAS studies. And using this hap map, so far more than 10 publications are already out, and this is really doing a good job currently. And it's all on public domain. You can actually get access to this particular hap map. So apart from this, you would also, so that was high density array. So you would also need something called as mid density panel because the cost of that array would be might be on higher side. So we have actually joined hands with Intertech, and we are trying to develop a mid density panel consisting of about 5,000 SNPs, genome-wide SNPs, which can again be routinely used in different molecular breeding programs like MABC and also in trait mapping. And taking the work further down, we have also developed something called as SNP-based QC panel. So now from the same 32 million SNPs, we developed high density and mid density panel. So we have further taken down the number of SNPs, and we have reduced the number of SNPs to just about 30. So these 30 SNPs can differentiate 99.9% of the breeding lines in germplasm coming from the Asian breeding program. And these lines can be used not just for fingerprinting or typing of the lines, they can be used in breeders program, especially in finding genetic integrity of B by B and R by R crosses. Because in Pearl Millet, you have these two major heterotic pools. I'll come to that later. So you don't need to contaminate B line pool and R line pool. And using this system of marker, you can really take out duplicates. You can take out anything which is contaminating the pool, and you can also find out the genetic integrity of the B by B lines, especially sister B by B lines, where your phenotypic scores would not be able to differentiate, and your most of the markers would also turn monomorphic. And we are also thinking to take this down to produce something called as digital seed ID by generating barcode. And we can actually track the purity of the lines starting from the plant breeder's backyard, wherein he or she is developing these B and R lines, making crosses, developing segregating generations, fixing those lines, putting them in the pools they belong to, and then generating single class hybrids. So you can fingerprint A line, B line, R line, and the hybrid, and you can track this in all your seed production chain, starting from breeder seed to foundation to certified to downstream farmers field as well. So this 30 SNPs are a wonderful pack of SNPs which can actually help track varietal integrity in terms of its genetic component. Now coming to trait mapping. So in Pearl Millet, we have mapped about 25 traits. So these traits related to yield parameters like flowering time, plant height, panicle length, seed wet. I don't want to get into details of that. On other side, on quality traits, we have both grain as well as forage quality traits. So iron and zinc content, IVDMD, which is in vitro organic matter digestibility, metabolizable energy, and things which are related to quality of forage. In terms of biotic constraint, downy mildew resistance. So downy mildew is one biggest constraint, and so it's very interesting. If you ask me to list, 3 important constraints of Pearl Millet, 3 important biotic constraints. So I would say downy mildew, and downy mildew. So it is extremely important constraint because the pathogen has got both spatial as well as temporal variability. And next is blast, which is blasting us all around. So it was considered to be a disease of minor importance in Pearl Millet, but since the last 7-8 years, this disease has become major, and the number of donors are extremely low at the moment, which is really a worrisome situation. And when it comes to aviarity constraints, we have terminal drought tolerance and traits related to nitrogen use efficiency. And on other traits, we have heterotic gene pool and stuff related to general and specific combining ability. Now coming to the QTL mapping work we have done for iron and zinc. So here in Pearl Millet, and also I think in many other crops, especially in rice, fortunately the iron and zinc QTLs are co-localized. And it's for the simple reason that the transporters for iron and zinc are non-specific. So the transporter which binds to iron can bind to zinc as well because of its ionic radius. And it's exactly for that reason you see these QTLs as co-localized. So that means if you select for iron, you select for zinc as well, and vice versa. And these are all large effect QTLs which have been validated, and these QTLs have been put in onto Intertech platform, which is a third-party high-density, high-throughput genotyping service provider. And the details are put on EIB platform. So if anyone wants, they can access the information of the markers and get the material genotype for iron and zinc. So apart from this, we also used another mapping population where we found iron and zinc, again co-localized, but on a different linkage group. So all these things are actually in place, and they serve different purpose. We also attempted some assertion mapping for iron and zinc, and we found some common markers across our biparentals and assertion mapping results. So this is work of one of my PhD students. So we tried to connect the story from biparental mapping to transcriptomics. So we found many genes which were housed in the QTL interval, which we had mapped from biparental mapping population, and they were found to be differentially expressed. So these some of the findings we published in scientific report and other journals. Those of you who are interested can actually contact me separately. Now, coming to the negative and positive selection side, so using the markers I just described. So I described this particular marker, which is on linkage group 3. So this is being used for negative selection, and this particular marker, which is from linkage group 1, is being used for positive selection. So it's like this. So the donor 863B has got not a very high level of iron and zinc compared to AIMP 92901, which clocks iron and zinc, iron on other side of 110 ppm, whereas in 863B it is around 78 to 80 ppm. So what we do is we use the panel of this marker to discard poor iron and zinc lines using the marker. So we can do negative selection. And you might know in Pearl Millet, for any variety, any hybrid to be released, it should have minimum levels of iron and zinc, 42 ppm iron and 32 ppm zinc. So using this marker, you can actually get rid of all the poor performing lines. And really, by doing this, you can exercise very low selection pressure on your rest of the population, which actually can be used to select many other traits of agronomic importance, because iron and zinc is important, but it's not everything, because there are many other traits which actually a plant builder would like to select. So if you apply very high selection pressure, you'll not be left with many number of individuals for excising selection for other traits. Now coming to positive selection, using the linkage group 1 marker, we can actually select also lines which are high on iron and zinc on either side of 100 ppm, the stuff which you see in red. So this will put a lot of selection pressure, and you will be able to get rid of much of the material which are low on iron and zinc, and you'll be able to retain only those high iron and zinc lines for your specific breeding activities. For example, if you already have some good combining ability lines, and you want to put these markers over there, so these can be used through MABC, and this can also be used in various segregating generations to get hold of lines which are exceedingly high in iron and zinc. Now coming to important abiotic constraint, nitrogen. So as I mentioned, Pearl millet is traditionally grown in areas which are low in nitrogen and phosphorus. So in the study, we concentrated specifically on nitrogen because in India, between nitrogen and phosphorus, it is actually nitrogen which is more limiting, whereas in Africa, in fact, it is phosphorus, or both nitrogen and phosphorus become pretty limiting. So this was a BBSRC defect funded project which lasted for about 4 years. So in this particular project, we in ICRISAT, those of you who have been to ICRISAT, we carried out this experiment in precision field. So in ICRISAT, we have red precision soils and black precision soils. So here in these soils, you can carry your experiments at greater precision because the heterogeneity from soil would be at the minimum, and the genotypes will be able to express better, and there will not be any kind of noise which actually comes from the soil. Allow me to sip some water. Sorry. Yeah. So in this, we took depleted precision field, which was depleted on nitrogen. So this field was not used. It was left fallow for about 5 seasons, and then we used that particular field. So using minimal nitrogen levels, we started this. So we had 3 treatments: N0, N50, and N100, using the diversity panel, which I just mentioned in the past, which represents a genetic diversity of 27 different countries, and it has been resequenced. So this panel was used along with some other lines, and we found a clear-cut difference between N0, N50, and N100. And we also find, interestingly, some of the lines which were pretty plastic, which means the yield was not much depressed between N0 and N100. So these are called as nitrogen insensitive lines. So we did some GWAS analysis as well using 2-season data, and we got hold of some good signatures. But we are in the process of further examining these signatures and trying to see if these are really the candidate genes for nitrogen use efficiency. So to take a complementary approach, we also followed the transcriptomics route, and we had all these important genes related to signaling pathway, relating to amino acid biosynthesis, especially this alanine amino acid transferase gene, and this glutamate synthetase gene. So we had used all these class of genes, and we had done some transcriptome analysis. So we found some of the genes here, which actually correlated here in the Welcome plot. So we are further trying to validate these genes, and if validated, we will try to put them in breeding programs, and these genes will have a lot of relevance in materials which go to A1 zone in Pearl Millet, which is supposed to be a dry zone of Pearl Millet where rainfall is less than 400 mm and soils are shallow, and this is mostly in Rajasthan and also parts of Gujarat and Haryana. Now coming to the biotic side of the story. So as I was mentioning, downy mildew has been an old age enemy for Pearl Millet, and there is a constant race between the breeder and genomic scientist and on one side and the pathogen on the other side. So this particular pathogen, as I mentioned earlier, exhibits enormous spatial and temporal variability, which means a resident source which is resident today can go susceptible 2 years later, and a resident source which is resident today here at Hyderabad can go susceptible maybe somewhere in Maharashtra in the same year. So and this recombines sexually, and it gives rise to a lot of new pathotype isolates. So it becomes really very difficult to match, and it is exactly for this reason no single cross hybrid would last for more than 4 years in farmer's field before it is defeated by downy mildew pathogen. So when we were mapping in the past, we were finding all small additive effect QTLs, but then we changed our strategy, and in association with our pathologist, Dr. Rajan Sharma. So this is work of one of my master's student, Durga Raju. So we, for the first time, identified QTLs which were large effect QTLs. The typical R-square value for downy mildew resistance would not be on the other side of 10%, mostly within 5 to 7%. But here in this case, we found QTLs which were explaining R-square, which was on the other side of 50%. A similar case we had in rest resistance. So this was work of one of our PhD students, Supriya. So we found a major rest resistance locus located on linkage group 1, which explained 69% of phenotypic variance. Now, coming to BLAST, as I was mentioning, so all these things were actually there with Pearl Millet since time immemorial, but BLAST is one new biotic constraint which is blasting the industry all around. And those of you who are working in Pearl Millet will realize how important this particular disease has become, especially when we are lacking on the side of proper donors. So we in the annual meetings, this trait was ranked as the most important trait. and a team was formed, and all of us, joined hands together to map blast-resistant genes and put it into the breeding pipeline. So we followed both forward as well as reverse genetic approach. So in forward genetic approach, we generated a biparental mapping population using lines which we got from different locations. We had data coming from the ECRIP centers, which we took about 4 to 5 years of data and found out the genotypes which were kind of consistent in terms of resistance levels. And we also did this genome-wide association studies using the same diversity panel. And we are doing QTL mining and comparative genomics. And to also complement, we are developing TILling genetic resource, reverse genetic tool to map, to mine the genes responsible for blast resistance. So these are some of the populations which we have generated. I don't want to get into details of that, but the mapping strategies which we are trying to follow is and this QTL-seq or bulk-seq approach, because obviously we don't have time to take it to F9, F10, because in Pearl Millet, even after 10 generations of selfing, there could be a lot of residual heterozygosity. So we just want to follow a QTL-seq or bulk-seq approach using F3 families to get hold of the genes and markers. And this is about the reverse genetic approach I just mentioned. So this line ICMR 11003 is supposed to be a reasonably resistant line in terms of blast. And this line was selected to generate TILling population. And we are at M2 stage. And we will follow TILling by sequencing to fish out genes for blast resistance. Now coming to something which will be of eminent importance to the plant breeders. So we published a paper last year in Frontiers. This was work of Dr. Mukesh Shankar, who was a sandwich student between me and Dr. S.P. Singh of IARA. So here in the study, for the first time, we found out more than 28 lines which had higher levels of blast disease resistance compared to the standard checks we were using. Standard resistant checks we were using. And this actually came from 5 environment data, including 2 locations, which, in fact, all 3 locations which are hotspots. So Delhi is supposed to be hotspot of blast. So is Bawal, which is in Haryana, and also Jaipur. So we did some GWAS for blast, but we are still in the process of validating the results. But then we also want to move a step ahead, and so we want to get into haplotype-based breeding. So we want to actually capture all the haplotype for blast resistance, and thanks to the data which has been generated already on the World Association Mapping Panel. So what we have done is we have actually used the resequence data. So we have recalled the SNPs using the new reference, platinum standard reference, genome. And then we have done this haplotype analysis. And in this analysis, if you can see, we actually found 4 haplotypes. So we did this haplotype analysis. So this HAP1, HAP2, and HAP3, if you see. So these haplotypes, they come from the World Association Mapping Panel. And HAPW is the haplotype which actually comes from wild species. Now, if you see, HAP1 is same as HAPW. So that means, if you jump to some wild species thinking that it might actually give you new set of genes, you might actually be wasting time. So while on one side we recognize that these crop wild relatives are reservoirs of important crop genetic diversity, we should also be cautious that we don't end up bringing the same gene pool which actually is present in the cultivated genetic background. So this is about the genetic variations which we did for the lines coming from East Africa. If you can see and follow my mouse, so these are the wild species of East Africa coming from the Sahel region, and the lines in yellow are the wild lines of the Western Sahel, and these are all cultivated lines. You see here. So, clearly there is a kind of structured diversity between the cultivated lines coming from different regions of the world and also the wild species coming from different regions of the world. Now coming to the downy mildew, I was mentioning, so we just wanted to examine the winning ways of this particular pathogen, because this pathogen is so interesting. I mean, it defeats all the deployed resistant genes within 3 to 4 years of time span. So we just wanted to see what exactly actually lies in its genome. So when we did this genome sequencing work in association with our national partners, Dr. Chandra Nayak, he is a professor from University of Mysore, based at Mysore. Dr. Shekhar Shetty, many of you might know, so he was his predecessor, and with him we started actually this work, but he retired, and it was Dr. Chandra Nayak who actually took the reign of the work. And we had in this work Dr. Sitara Satvati, who is the project coordinator of Pearl Millet. We also had Dr. Ratan Yadav, based at Bristol University, UK. So we had a big team, and so to publish this paper, we had a tough time because the genome size was not on the expected grounds. If you see all mycetes, class of fungi, the genome size is between 40 to 50 MB. Whereas in this particular case, in Sclerospora graminicola, it was almost 300 MB. And we had a tough time, so we generated flow cytometry data and we provided all the evidence before the paper got accepted. But interesting thing here is, you see the number of genes. So I think in Pearl Millet, I didn't put that slide out. In Pearl Millet, you just have about 37,000 annotated genes. It's such a big genome, 1.8 GB. Whereas this has got just about 300 MB, genome. And it has got more number of genes. You see here. Over 65,000 genes. So this definitely shows that it's a pretty intelligent pathogen, something like, SARS coronavirus. So this particular pathogen actually is smart and is able to track the R genes which are deployed, and it modifies its AVR gene and then matches, and then it defeats it. So this is going to be a very interesting model to study, and I'm sure if you are able to decipher fully how exactly the pathogen is able to learn and is able to adapt, we'll be able to take the findings to other crops as well. So we did some sentinel analysis as well, and this was all on expected grounds. So coming to the holy grail of plant breeding, which is genomic selection nowadays. So genomic selection, everyone is talking about in plant, but it is mostly in self-pollinated crops. So their genomic selection is pretty easy because the line you select, on the basis of performance becomes your final line, which goes into the release system. Whereas here in genomic selection program for a cross-pollinated crop, there is very little correlation between performance and the hybrid performance. Those of you who have worked in hybrid crops would know this. So here, if you have to run the genomic selection program, you have to run this, you have to train your model both on the R line side and also on the B line side, so that you train the model and generate your heterotic gene pool on the basis of GEVs, on the basis of genomic stirred breeding values. Now coming to something I was talking in my initial slide on health-related benefits of Pearl Millet, especially low GI. So so you might know diabetes is prevalent all around, and especially in Southeast Asia and in India. I'm sorry. In India, it is almost India has become diabetes capital of the world, wherein every fifth individual is either diabetic or pre-diabetic. So we just we had some clue, but we just wanted to check our hypothesis, and we then followed the words of wisdom of Hippocrates, who said, "Let thy food be thy medicine." So we looked into the genetic variability present in Pearl Millet grains for two important classes of starch. So if you see starch can be fractioned into three components. One is readily digestible starch, which gets absorbed in the stomach within first 60 minutes. Then comes slowly digestible starch, which actually is absorbed between 60 and 120 minutes. And then comes resin starch, which is absorbed after 120 minutes or is not absorbed at all. I mean, it passes to large intestine and it becomes food for the gut microflora, where it generates a lot of important, vitamins for the body. So we had this project with our UK partner, Praswat University, and this was funded by BBSRC DFID. So in this particular project, we actually profiled lines for SDS and RS, and then generated single cross hybrids. This actually is Bawal location, which is close to Delhi, and it is an important even zone location for Pearl Millet, dry zone location for Pearl Millet. And in this location, some of the hybrids really, performed well, not just for the target trait, which was low glycemic index, but also for other agronomic traits, because we understand that these traits are silent traits. Unless and until we pack it into agronomy, it will not be, taken forward. And these hybrids are actually being taken forward currently with help of our industry partner. Now coming to molecular breeding product. So Dr. Rao mentioned this briefly. ICRISAT actually takes pride in development of some mega hybrids. So this includes HHB67. So HHB67 improved was the first molecular breeding product released in the year 2005 by Dr. Tom Hash and partners, along with CCS HAU Hisar. Now, after a good number of years, I mean, it stood the score of downy mildew for a long period of time, over 19 years, and then it started showing signs of susceptibility. So we knew that this is going to happen. So we had started this program, and we actually pyramided downy mildew resistant scutales from 2 different donors, and 3 different downy mildew resistant scutales were introduced in the male parent of HHB67 improved, which was released way back in 2005 by Dr. Tom Hash and CCS HAU. So we introduced 3 downy mildew resistant scutale and made it more resistant to downy mildew disease. And by doing this, we also actually increased its grain and fodder yield while decreasing the crop duration. So what happens is, whenever you do this, the crop duration gets extended, as it happened in the first cycle. In the first cycle, the crop duration got extended by 1 day. So in this particular cycle, it got again reduced by 1 day. So we have balanced it off. But though the days to maturity has got reduced by 1 day, but the grain yield has got increased by 16%, and the dry fodder yield has got increased by 23%. So in terms of absolute values, this amounts to about 400 kgs of grain yield per hectare, or about 980 kgs of dry fodder per hectare. Now, if you multiply this by 850,000 hectare over which HHB67 improved is being grown, and take it on the value scale. If we assume, if we take the minimum support price, which is 2,250 rupees of Pearl Millet, and about 750 rupees per ton of dry fodder, rate, the total value actually comes to on the other side of $150 million US at the current exchange rates. So this is the magic of investing in high-end science, which actually leads to downstream impacts. Apart from this, but before I go to Marusona, so this HHB67 Improved 2 was actually dedicated to nation by Honorable Prime Minister of India on 28th of September 2021. And now it is in the process of entering into the seed chain, and we will make sure that it actually goes much beyond 10% area. In fact, our target is to take it to about 30% of the total Pearl Millet area. The second molecular breeding product I would like to talk about is GHB 538, which is released as Marusona, which translates to desert gold in the state of Gujarat. And it is an improved version of GHB 538, which is again an extremely popular Pearl Millet hybrid, which has gone down to downy mildew. And it is the first marker-assisted breeding product released by the state of Gujarat. So these are two molecular breeding products which are pretty important in terms of its impact and in terms of returns on investment. Now, finally coming to some nuts and bolts. And so we've been talking about all these things, but then this is the famous, breeder's equation. So here, the rate of genetic gain, the rate of genetic gain is a function of heritability or prediction accuracy. And also it is a function of selection intensity. And this actually can be increased if we do better on genotyping cost and its estimation of breeding value over phenotyping. And also most important is the L, which is the length of the breeding cycle. So if you see on numerator, there are 3 terms, whereas on denominator, there is only 1 term. So obviously this has got the highest effect on the rate of genetic gain. So if you actually can increase the length of cycle or decrease seed to seed duration between your generations, then your genetic gains is going to jump. And in this direction, I think ICRISAT has already taken a first step. So you must have seen Honorable Prime Minister of India when he visited ICRISAT a few months ago here. He inaugurated this climate change facility and also rapid gen facility. So what I'm showing you is the picture of rapid gen facility, wherein we try to squeeze the duration between seed to seed and decrease the L so that rate of genetic gain can be actually increased. And for Pearl millet, we also have these dark, overhead covers, which actually can help it set seed at much lower, time periods. And we are also trying to manipulate the quality of light in terms of its wavelength and intensity, and also related to photoperiod, soil moisture, and the nutrient content, especially nitrogen. So if you starve your plants on nitrogen, it flowers pretty early. But then if you starve it too much, it might not flower at all. So finally, this is going to be my final slide. So coming to future threats, so we have to work both on the plant side as well as the pathogen side. So when it comes to pathogen side, as I mentioned, both for blast as well as downy mildew, especially in crops like pearl millet, you have to really see how exactly the pathogen is evolving. You have to make sure to read the way pathogen is learning and pathogen is evolving, and you have to take the clues out on the basis of genome sequence information we have generated, and also on the basis of other mapping exercise we do to find out the respective AVR genes which are present in the pathogen, to put them and make durable sources of resistance deployed in the breeding program. And on the plant side, your genetic resources, of course, are your strength, starting from the collections coming from gene bank, because it is the gene bank which actually holds the allele diversity for a particular trait. So well characterized germplasm collections are the starting point, and then you can have your forward and revert genetic approaches, and then you can have enabling technologies. So genomic technologies are one, but you can also have gene editing technologies and other high throughput technologies. And this would take us to the next generation breeding by pyramiding new useful alleles, and take us into the real era of genomic selection, which so far has been only on the animal side. So with this presentation, I thank all of you for your patience, and I'm open to questions. Thank you very much.
Thank you, Rakesh. Now the floor is open for questions. I think Swapnil Bharati, you raised your hand. Can you please ask your question? Unmute. Swapnil Bharati, are you there? I think you need to unmute and then ask your question. Okay. Swapnil is not asking the question. So fine. Anyone else has any questions? Rakesh, can you close your presentation?
Yeah, I'll just do that. I'm just trying to figure it out. Yeah, just a minute.
Rajiv, you like to ask a question?
Yes, Aparna, yes, if I could please. Thanks a lot for the brilliant talk, Dr. Srivastava. My name is Rajiv Thakur, and I have two questions. One is, regarding the, it seems you've been working also on the transcriptomics bit, and now, as has been seen in the human models, there's a component of variation, variance, which can also be explained by the epitranscriptome. So I'm not sure how much of this is being transferred to the crop genetics, and I wonder if at ICRISAT you guys are also looking at the epitranscriptome, if some sort of variation can be can be explained by the epitranscriptome also. That is my first question. And the second question I would like to ask you is, if you could go back to your slide on genomic selection, and it seems you did you tried to find out how GS could sort of decipher heterosis in Pearl millet, but it seems the results are not very conclusive. So if you could go back to that slide and could please explain us a bit more how much genetic genomic selection made sense in finding out the correlations with heterosis. So if these two questions could please answer. Thank you.
Yeah, so Rajiv, thanks very much for your observation and your questions. So your, I think, point is very valid that this epitranscriptome or epigenomics is an important area where we should be looking for. And we are in fact doing this. We have already taken a step in this particular direction. But it's like, see, all of us have got limited pot of money to carry out our research. So while what you mentioned is pretty exciting and it makes a lot of sense, but then what we do is we follow the normal sequence. For example, if you are able to find a reasonably good source of variation within the genomic side of the road, then we don't go into epigenomics. We get into epigenomics only when we don't find meaningful sources of variation on the genomic side. For example, there are some traits. I didn't talk about striga. I deliberately took the slide out because I thought audience would be from India, so I took it off. But then we are doing a lot of work for striga with help of our West African partners. So in striga, unlike sorghum, in Pearl millet, the variability is very less. The genetic variability is very less. And it is related to the way Pearl millet has actually evolved in West Africa. So what we are doing is then we are trying to get into the other side of the road, which is of course epigenomics, but also we are trying to get into gene editing. So we are trying to find out some kind of candidate genes using using comparative genomics, especially from sorghum, and then trying to see if those genes actually are making sense on Pearl millet side. But yes, this epigenomic side point is well taken. It makes a lot of sense, and we would love to do that, especially for traits where genetic variation is not found. And I think striga is one trait where we are trying to go on the other side of the road. And if we get some good insights there, I'll be very happy to share this with you, Rajiv, on epigenomics. On other ICRISAT crop, not in Pearl millet, but in pigeon pea, a lot of epigenomics work has been done. Yeah, so if you're interested, maybe on a separate one-to-one window, I can actually let you know the story on epigenomics side. But then, as I mentioned, when we all are pragmatic, we all have a given pot of money, so we just go this way. Now, coming to the genomic selection, I actually did not show the slide because, I thought maybe I was given time of 1 hour, so I didn't put that slide on heterotic gene pool, which we had developed. So when we developed this heterotic gene pool, we could see not just B and R lines are two different heterotic gene pools, but within B and R lines, there are specific heterotic gene pools. And when you superimpose this heterotic gene pool information in your genomic selection model, then only it starts making sense. Because when you make your training set has to be of that particular heterotic gene pool coming from that specific B by B cross, which actually mixed with your R by R cross. Otherwise, you are not going in any direction. So obviously, this genomic selection exercise cannot move forward in cross-pollinated crops, especially for single cross hybrids, unless and until you superimpose your heterotic gene pool, information there. Rajiv, I hope I have answered your question.
Yeah, thanks a lot. Definitely, I got some, especially the sort of work you are doing in West Africa regarding the striga. So that definitely shows, where exactly to look at for these answers. But thanks a lot. Thanks for this, brilliant. Thanks. Yeah.
Rakesh, there's a question from Aishwarya Thakur. And the question is, any high folate lines were identified? Kindly give the range of folate content in polished and unpolished grains.
Yeah, that's a very good question. I'm sure the background is either rice or some other cereal, because in pearl millet, you really don't polish the grains. You just remove the husk and, it becomes fit for consumption. So the good thing is, the aileron layer is not removed. Just like in rice, if you do more polishing, you lose all the precious vital, nutrients. And coming to the range, so range is huge. It varies from as low as, so we compare it with respect to rice, because in absolute terms, what happens is there are many parameters which actually can jack up or jack down the values of folate content in pearl millet. So what we do is we keep rice as standard, and then we express over rice what percent we have got. So this is how we actually do. But if you are interested in finding out specifics, I can provide you. In fact, I can share you the data for folic acid, and I can tell you, there is a lot of variation, and a lot of it is actually heritable.
Is there any other questions, please?
Hello, sir. Good afternoon.
Yeah, this is Vishal Vani. Go ahead, please.
Sir, we all know the bajra or the pearl millet is like a tiller producing crop. But in case of the tiller producing, we facing the problem like the some up downs in the tiller producing or the synchronization in the bajra. Can we change this process by using the genome selection or any other method to reduce the losses in summer crop or in the major of our kharif crop due to losses of the seasonal birds to damage the early matured heads?
Yeah, Vishal. So that's a very important observation, Vishal. So of course, this tillering is one trait which is two-edged sword. It is good as well as not so good if you see from a particular perspective. If you see from the perspective of crop, so tillering basically is an adaptation trait. It will make sure that the plant is able to survive. And if you see into the genetic basis of this tillering, this is basically more in the lines which come from dry areas where rainfall is erratic. So they produce tillers so that they can actually catch up or they can spread the risk. But when it comes to modern plant breeding activities which actually help you design your plant type which are for assured irrigation types or where a lot of input is actually given. So there you don't want tillering because it actually becomes a kind of noise in your stuff. So of course by doing tillering is a highly heritable trait, and genetics is not also very complex. So you can easily get away with this tillering trait. And if you see all these hybrids which are coming from private sector, they're almost like sorghum. They have transformed pearl millet to sorghum. They have strong stem, and they don't tiller. They will just have one or two, three tillers, and they would have strong panicle on it. So it looks pretty attractive, especially the hybrids coming from Corteva and Bayer. If you see, they almost look like sorghum.
Rakesh, there are two more questions. One from Shashi Kumar. The question is, in which linkage group the blast-resistant genes are mapped in pearl millet?
That's a very interesting question. So had you asked in which linkage groups blast-resistant genes are there, I would have answered easily. So linkage group 4 is supposed to be a disease resistance rich linkage group. This corroborates to pseudomolecule 4 or chromosome 4 in the current genome assembly. So most of the disease resistant genes, blast, downy mildew, rust, actually rest over there. But then small effect, QTLs are present all over the genome, but we really don't bother about them. We just bother about the ones which are on other side of say 10%. And in this case, fortunately, blast resistance, the genetics is very simple. It's governed by a single dominant gene. So we can actually target the major effect, QTLs and put them over there.
Okay,
that's what actually makes sense.
Yeah, and the next question is from Shankar Yadav, and he's asking: is there identified QTL combination which control downy mildew?
Yeah. In fact, many. So as I mentioned in some of my slides, we have race-specific, QTLs. And the important thing is, if you put a QTL which gives you resistance against race A, and also put a QTL B which gives you resistance against, or QTL 2 gives you resistance against race B, then the individual effect of these QTLs is fine. But then if you look at the combined effect of the QTLs, it is more than the sum of these individual effects. So if you pyramid QTL 1 and QTL 2, so it will give you resistance against not just pathotype isolates A and B, but might also give you resistance against other pathotype isolates. So that is why we actually pyramid, more number of QTLs.
Yeah, Rakesh, two more people have raised hands. I think Swapnil Bharati had raised earlier. Now again, so can you ask your question? Unmute yourself, Swapnil. If not, Suren, can you go ahead with your question?
Hi, Rakesh. First of all, thank you. Great presentation and a lot of work. Part of that work, of course, goes over my head, but I know how that work can be used by a breeder. So very, very good work. So I have a comment and I have a question. So first, I'll say the question. Question is about, you showed that iron, zinc, it's kind of a pleiotropic effect or something like that, because you can select one and you get the selection for the other through the molecular markers, right? Yeah. But with that nutritional quality selection with markers, has there been any study with any negative impact of yield on yield? Is there any? That's my question. The comment is on that 5G slide. Of course, one cannot find mistakes with Rajiv Varshni's work, but I am wondering, why do you keep 1G as genome? Genome came later, germplasm came first. So I feel there should be a reversal of that 1 to 5. That's just a lighter vein comment. So, but please let me know about my question.
Yeah. Thank you so much, sir. So first of all, your question actually is taking me way back 18 years, wherein you and I were sitting in the headquarters of Syngenta Seed, and you were interviewing me. So yeah. It's making me nostalgic. So, sir, thanks very much for this opportunity and for all your help and support. Because though you said many questions got over your head, but then let me tell you what I am today is all because of the training I received from you. So that really gave me a good jump start to take things here in ICRISAT. So thanks very much, sir. Now coming to your question, sir. So iron and zinc, these are not pleiotropic. So you can find lines which are high in iron and low in zinc, and vice versa. And this could be due to many factors like competitive inhibition. Though the transporters are same, they've got same ionic radius, but then maybe there is some other mechanism which is working in the background which makes iron and zinc go independent in some of the lines. So you can actually break the linkage. So you can have lines, and we do have such lines in our real mapping populations, wherein you can find lines which are high in iron and low in zinc, and vice versa. And you get to see such lines even in the breeding program. But when it comes to this specific QTL, so that QTL specifically, that locus actually allows you to do a kind of co-selection for iron and zinc. So these are two different things. Now, coming to, I'm forgetting. So what was your question?
My question is that, so those QTLs probably are at different chromosomes, but they are co-selecting the two
Right.
nutrients, right?
Right,
Anyway,
right.
I was asking, is there a, because you talked about some other nutritional quality also, is there a yield penalty, which
Yes,
generally we have seen in maize and other
Yeah.
crops?
Yeah.
Is there a yield penalty?
Yeah, so I think a lot of studies on this were done in the past. In fact, when this program was taken up by Harvest Plus, so at that time, a scientist called Dr. Govind Raj, who now is with Harvest Plus. So he actually had done extensive studies on this, and we had concluded that there is no significant, there is negative correlation between iron and grain yield, but it is not significant. So that means if you generate a big segregating generation population, you can always break the linkage between high iron and low yield.
Sir, could I join in, Tiku sir? Could I join in?
Yeah,
Yeah, go ahead, Rajiv.
Sir, sorry, Dr. Srivastava, I mean, I'm jumping in. Sir, I mean, I think what you talked about, the iron and zinc, getting together on the chromosome to get their being closely placed. What I feel is also there's a pretty established theory by now that the genes which which act together and which have some sort of correlation in function also, they tend to go very close by each other. So all the tightly linked gene, and we know that in humans that when we want to go for iron absorption, we also need zinc. So I think in the physiology of zinc absorption and iron absorption, both of these traits are pretty linked, and that is probably one reason why the traits which are linked together, they, it falls in the same hypothesis that they tend to be tightly linked as well. So little wonder that we see the genes for the iron and the zinc uptake or efficiency, they are being, they are being transmitted very closely together in heredity. And yes, that's true that in certain cases they are on a different chromosome, but if we look at the haplotype as such, I think they both are tightly linked, and they're linked for a reason. Sorry for jumping in.
No.
Yeah, that's fine.
I think that's a valid point. Yeah.
So that's a very valid observation. Yeah, that's a very valid observation. It's the way nature actually allows co-evolution of crops with human beings.
Yeah, exactly. Yeah,
because unconsciously human beings in the past have done these kind of selections which have taken things this way, because the set of genes or the set of genome which we see currently, it is all a result of either environmental selection or human selection. So consciously or unconsciously we've been doing this selection, but maybe we have never understood what exactly is the complex background mechanism which is operating.
Swapnil Bharti, last chance for you to ask the question.
No, Dr. Rao, I am yet to comment on Dr. Tiku's second question, which is the GS, the comment. So sir, Tiku sir, if you see, though I presented that slide of Rajiv Vashne, but I read out things in the right sequence.
Yeah. No, I was in a lighter vein because yesterday in Bangalore, also somebody showed that slide.
Okay.
And it went through the same sequence but then it didn't. I was talking to somebody and I said, why do you, genome was discovered much later, germplasm came first. Yeah. Anyway, sorry, that was.
This is the effect of your training, sir.
Thank you. Yeah.
Okay. If there are no other questions, I think we are almost running towards the end of the session. First of all, I mean, I'll use the chance that I have become, of being a moderator in this session to just make a comment on Striga, which Rakesh, you mentioned. I think Striga is a very, very dicey, very slippery kind of pathogen. You said that in India it's not that serious. And actually, what the farmer does is that he changes from crop to crop when he or she gets the Striga occurrence. And then the damage is not seen because on the surface, the Striga appears once in a few years, but then it does the damage because it is subterranean in nature. And West Africa, the Striga is very big, Harmatica is very big, very visible, very colorful, so therefore it gets attracted. But then I've seen Striga, in fact, way back in the early 80s, did produce a paper on the loss due to striga in sorghum. I think that could be a good indicator of what it is. And I know there are a lot of instances where people have lost the crop, and once they, for example, they lose sorghum crop, they go to maize. And I've seen striga asiatica in maize, in sorghum, in pearl millet. So therefore, it is there. It is just for you to recognize and keep working on that. And with that comment, I would like to take this opportunity of summing up the presentation. Rakesh did a fantastic job in just 1 hour of summarizing decades of work, and there's a stamp of quality, ICRISAT quality, and Rakesh's personal quality in the presentation. He started off explaining why genomic techniques and technologies are important with reference to the global population and increase in global food production, increase requirements. Then he went on to describe pearl millet and its importance. Especially a couple of things that I noticed was its ability to germinate in very high temperatures. Of course, pearl millet is a crop highly adapted to high temperatures. So obviously one part should be the seed germination, but that's very interesting. And then designer eggs. I think, we know that maize got importance, and especially the orange types got importance because the growth of maize is in India particularly is parallel to the growth of poultry industry, all because of its carotene content, which imparts yellow color to the yolk of egg. But here is one more crop which can give not only a solution for diabetes but also for other heart-related diseases because of its change of HDL-LDL ratios. So it's a very interesting observation and future opportunity. And it touched upon the selection based on eye selection, which is a conventional plant breeder, versus data-driven selection by a new gen plant breeder who base their, who make their selections based on data and detailed data and data analysis and then interpretation. And I believe that the human, the data-driven selection is based on artificial intelligence, whereas you still need the natural intelligence of a human being to interpret all that and make the right choices. So there is still, it's all a gray area, but the movement from eye selection to, which is based on phenotype, to data-driven selection, which is based on genotype, is a transition in which we are all living at this point of time. And with the new technologies coming up, that will also become finer and finer. And then he went on to describe the genome sequencing in case of pearl millet and how they have come down from 32 million SNPs to just a few SNPs which can be handled very effectively in plant breeding programs. And then interesting aspect on genetic barcodes and which can be related to seed quality systems. I think that is where digital seed identity and genetic identity. One of the problems that plagues the seed industry is the seed quality tracking, especially in that the seed identity, the genetic identity of the seed. And if there are new technologies coming in, you're not only helping the plant breeder, but you're also helping the seed industry, which has a much bigger, larger implication to seed industry. And of course, the best part of your presentation was on the trait mapping, iron, zinc, nitrogen use efficiency, downy mildew particularly, rust resistance, blast resistance, which is a new disease. I think all that is very, very interesting and has huge bearing into the future of pearl millet breeding. Yeah. And of course, the taste of pudding is in its eating, and you did present about the successful deliveries from genomic selections, HHB67 improved 2, which has already gained a lot of traction in the market. And the very interesting part of giving a dollar value, 150 million US dollar every year. I think it's every year that you wanted to say, because of the, what was grown earlier, what is grown now, and what is the value it is bringing. And also the very interesting aspect that it is 1 day less in maturity. I think it's probably 1 day less in flowering, and that has even a higher value, especially when you are fighting climate change, and pearl millet is a crop grown in drought-prone areas. So 1 day early means a lot. That much less risk with reference to environmental vagaries for the plant breeder. And you did talk about the speed breeding and rapid generation advancing facility, and I think that is the future. Everyone wants, I remember Dr. Lee House telling us, this is way back in the early 80s, when he asked us for a report, and we said, when do you want this report? He said, everything in ICRISAT is wanted yesterday. So that is the pressure on plant breeder. Okay, so all the speed breeding facilities and rapid generation advancing facilities, I think they'll be very useful in future to improve the rate of genetic gains. Finally, I would like to thank you, Rakesh, for the excellent presentation. I think it is decades and decades of work by stalwarts in pearl millet, K. N. Rai, and S.D. Singh, and I mean Rajiv Vashne, and so many others who were part of S.C. Gupta. So there were many stalwarts who have laid foundation for this kind of wonderful work, and you're carrying on that legacy. And congratulations to you, and thank you very much on behalf of ATPBR for this wonderful presentation. Thank you very much.
Thank you so much.
Thank you, Rakesh.
Thank you. Thank you.
Thank you. Over to
Thank
you,
you, Dr.
Aparna.
Rakesh. Thank you very much. Yeah, thank you, Dr. Rakesh, for such a wonderful presentation. And Rao sir has summarized it very well, that how informative and how information was kept in a nutshell. And it was an opportunity for here because such crop is not very commercial in terms of like chili and tomato. So I'm sure a lot of people would have learned a lot. Thank you, Rao, and for summarizing the session. Thank you all the participants for attending the session. And before closing the session, I would like to let you all know that our next speaker is Dr. Ramesh Bhatt. He's the professor at US Harvard, and he's going to talk about the genomic aspect of peanuts. So another exciting session will be awaiting for us next month, and further information will be communicated to all of you about the date and time. So thank you all, and have a great weekend. Thank you all.
Thank you.
Thank you.
Thank
Bye-bye.
you. Thank you so much. Bye-bye.
Thank you all for joining in.
Thank you.
Thank you.
Thank you, sir.
Bye-bye.
Thank you. Bye-bye.
Bye-bye. Have a great weekend.